Hello friend, today we are going to discuss the plant growth hormone part 2. In our previous lecture we have studied two hormones, auxin and gibberellin. Here the third one is cytokinin. Cytokinin is a growth hormone that promotes cell division. Latham is the scientist who first gave the name cytokinin to this hormone, and first time it was discovered by Skoog and Miller during the investigation of nutritional requirement of callus tissue culture of Nicotiana tobacum. When they tried to grow this Nicotiana tobacum callus, then they wanted to find out the nutritional requirement. At that time, they observed that the callus proliferated when the nutritional medium was supplemented with coconut milk and degraded sample of DNA. Whose DNA that was? Degraded sample of DNA which is obtained from herring sperm. Here this herring is the name of a fish, and from its sperm the DNA material which is extracted, that DNA is called as the herring sperm DNA. Okay, and that was named as kinetin. Chemically, kinin are 6-furfuryl aminopurine. First natural cytokinin was obtained from unripe maize grain by Latham. Okay, remember this one. Then at that time this cytokinin was named as zeatin. Here the synthetic cytokinin is 6-benzyl adenine. This is the synthetic cytokinin. 7 different types of cytokinins are present in plant. Natural cytokinins are also reported from plant like banana flower, apple, tomato, and coconut milk. Now about the physiological effect and application of cytokinin. Besides cell division, it also promotes the cell enlargement. Okay, means it has it is involved in two functions or two processes: cell division and cell enlargement. Here the main thing is that high concentration of cytokinin promotes the shooting. Means if it is present in a high concentration, then the plant growth is more in at the shoot. And if it is present at a low concentration, if cytokinin is present in a low concentration, then it induces the root development. Okay, but high ratio causes bud and shoot development. If cytokinin is present more than auxin, then bud and shoot development takes place. Cytokinin and auxin ratio and their interaction controls the morphological differentiation. In this way, the morphological differentiation means formation of different plant part, that is based on this ratio, the ratio of cytokinin and auxin. If cytokinin is present more than auxin, then shoot growth takes place, and if it is cytokinin is present less than auxin, then the root development will takes place. Okay, then it promotes the growth of lateral bud and control the apical dominance by cell division. Cytokinin promotes the growth of lateral bud. If only auxin is present, then the apical growth takes place. But when cytokinin is present, the lateral buds, they are going to develop. This controls the apical dominance. Last time we have studied what is meant by the apical dominance. If only the apical growth takes place, that growth of apex inhibit the growth of lateral buds. Okay, this is called as apical dominance, and here this cytokinin controls the apical dominance by cell division. Cytokinin delays the senescence or aging and abscission process. Abscission process means falling of leaves in the plant organ. Okay, this was reported by Rickman and Lang in 1957. The formation of interfascicular cambium and expansion of cell are other function. This is also the function of what? Cytokinin. It helps in the formation of interfascicular cambium. Interfascicular cambium, which is present between the vascular bundle. Okay, suppose these are the vascular bundles. Interfascicular cambium is present here. Okay, in this way it also breaks the dormancy and promotes germination of seed. It breaks the seed dormancy and help in the germination of seed. Cytokinin reverse the apical dominance. In above topic we have discussed that one. It controls the apical dominance. Okay, it induces RNA synthesis and formation of interfascicular cambium. Here also we have seen that about the interfascicular cambium. Okay, this is all about the cytokinin. Here the next hormone is ethylene. Ethylene is a gaseous growth regulator. Okay, Denny reported ethylene is effective in a fruit ripening. Remember this, ethylene is involved in fruit ripening. Here again establish that plant naturally synthesize ethylene. Crocker proposed that ethylene is a plant hormone responsible for fruit ripening. Okay, that we have studied here. Fruit ripening. It is simple gaseous hydrocarbon and it has essential role in a fruit ripening. Most widely used compound as a source of ethylene is ethaphone. It is synthesized in root, shoot, apical meristem and ripening fruits. Okay, below this the physiological effect and application of ethylene. It promotes the ripening of fruit, as we have already seen, like banana, apple and in mango. It stimulates initiation of lateral root in plant. This is the another function. It stimulates the growth of lateral roots in plant, and it breaks the dormancy of bud and seed. Okay, that we have already studied. Then it accelerates the abscission activity in leaves. Abscission means when leaves falling down from the tree. This is called as abscission. Abscission of leaves, flower and fruit by forming of abscission layer. Abscission layer, how this abscission layer is formed? Suppose that this is a stem and here the leaf is there, okay, and because suppose in summer season it prevent the excess of loss of water, that's why the transpiration process, it has to, for minimize the transpiration process, it has to cut down the leaf, okay, then it forms the abscission layer here, due to which what will happen, the leaves cut off from the tree, okay, in this way it forms the layer abscission layer. Ethylene inhibit the growth of lateral bud. It inhibit the growth of lateral bud. Lateral growth will not take place. Causes apical dominance and retard flowering. What will with the result, it causes the apical dominance. Apical growth takes place, more and more apical growth takes place. Lateral growth inhibited by this one, and flowering is also retarded. The growth of flower is also inhibited by this hormone. This enhances the process of senescence of plant organ. It inhibits the flowering in most of the plant except pineapple. In pineapple plant, it will not inhibit the flowering. It causes epinacity, means dropping of leaves and flower, that we have already studied. It increases the activity of chlorophyllase enzyme, causing degrading effect in banana and citrus fruit means like orange, lemon, these are the citrus fruits. Last hormone is abscisic acid. It is a natural growth inhibiting hormone, okay. Ethylene also the growth inhibiting hormone. Abscisic acid is also growth inhibiting hormone. Kans and Endicott observed that the shedding of cotton ball was due to the chemical substance abscission first and abscission second, wherein isolate the substance from the bud of acer that can induce the bud dormancy and name it dormine, okay. Then these two identical chemical substances were given the common name abscisic acid, and it is synthesized in leaves, fruit, root, seeds. Chemically, it is 15 carbon sesquiterpenoid, and it is synthesized from mevalonic acid. Now about the physiological effect and application of abscisic acid. It promotes the abscission of leaves. It promotes the abscission, so it is abscisic acid. It induces dormancy in many plants. It controls dormancy in bud and seeds by inhibiting the growth process, okay. It accelerates the senescence of leaves, flower and fruit. It inhibits delay cell division and cell elongation. It inhibits, that means it inhibits directly growth, and suppress the cambium activity, means if cambium activity stops, the girth of the plant will not increase, okay, it remains as it is. In this way it inhibits the growth. It suppresses the cambium activity by inhibiting mitosis in vascular cambium. The abscisic acid could cause efflux of potassium ion from the guard cell and result in closure of stomata. Okay, this mechanism closes the stomata. This is known as anti-transpirant. It acts as a stress hormone by inducing plant to bear adverse environmental condition. Okay, it inhibit the flowering in a long day plant but stimulate the flowering in short day plant. In this way, it is the growth inhibiting hormone. Thank you, friends.